So in our continuing series about proteins, I thought it might be good to go and give you a little bit of background that you're going to be hearing about soon, and that's how proteins are made. So we've talked a little bit about amino acids, and those things we call amino acids are the building blocks of proteins. And so we saw that there were 20 of them that we're going to be concerned with, most of which we will get our bodies can manufacture, some of which we need to get through our diets. So when we eat foods that have proteins and they get broken down, in other words, those peptide bonds get broken down, and then we get the individual amino acids, then the individual cells, so here's a cell up here on the left, use those amino acids to construct other proteins that the body needs. So where a lot of that starts is if this is a cell, you're probably familiar with cells from your past biology or life science classes, and here within the cell is the nucleus, and within the nucleus is DNA. Right? We all are familiar with DNA, so DNA is a double helix. You can see it down here below in this spiral staircase kind of look here. We can see that along these rungs of this ladder of sorts are different colors, which are representing some things we'll talk about pretty soon. But what this is a code, and this is a code for proteins. So all of the DNA that you have in your cells is coding for various proteins. So that information has to leave the cell, so there has to be some information, something that tells the DNA that a protein needs to be made, and it's going to be a part of that DNA is going to get copied, and then it's going to ultimately get translated into this thing we call a protein. So this is kind of the way things go. We have DNA, which is going to make a little copy of something we call RNA. And that copy is actually a part of that DNA. And then it's going to leave the nucleus, it's going to go out, and proteins are going to get constructed. Now, when proteins are constructed, they are constructed, first of all, in a primary structure, which is basically this long string of the amino acids that get put together by dehydration synthesis into peptide bonds, which unite them, and they form these large things called polypeptides. And then we're going to go through what these levels of structure are all about one more time. So here's the primary structure, primary protein structure. And what you see here, all of these up here are representing various amino acids. You can see their abbreviations here, and that we can have a long chain of amino acids like this. And then here's a second chain, this B chain. And that these can be linked by sulfur. These are sulfur bridges, sometimes called disulfide bonds, to form something that is a little bit longer and has some functional relevance. So here, for example, is insulin, and it's made up of 2 chains of these polypeptides, and the order of these amino acids in these long primary structure chains is what determines the ultimate fate of this protein. So it has to be these amino acids in this particular order in order for it to be a functional protein. So if there's some error, if something gets replaced, wrong amino acid somewhere, then it can change the ability of that protein to function. And that can lead to all sorts of issues. So the amino acid sequence is what's primary. It's the most important thing to start. You have to have this amino acid sequence in the right order, right number of amino acids. And if there is some kind of a change, we could alter the fate of that protein. So here's the example that was used. So sickle cell anemia is one in which there is a substitution. There was a normal hemoglobin would have had at the seventh amino acid in the lineup should have this particular amino acid, glutamic acid. But in sickle cell, instead of glutamic acid, valine is the replaced amino acid. So sickle cell actually is not a good condition under most circumstances. We don't want sickle cell. And this is in that beta chain of hemoglobin, knowing that there's an alpha and a beta lineup of these amino acids, and this is the beta one. The reason why it's not good is because you can see that in this picture here of red blood cells, some of them have an odd shape rather than being the round shape that a normal blood cell takes. Some of them have oblong or sickle. This one here is very much a sickle shape. And the problem with those shapes is that they can't carry oxygen. And so in a person who has sickle cell anemia, depending on the severity of their condition, they might have some of these cells might have sickle cell, and some may have a normal shape, in which case they're going to experience some of the symptoms of sickle cell, and we'll look at that actually coming up. So once you get the long chain of amino acids, then you get that chain starts to make little folds in it and starts to change in its structure, and that's due to hydrogen bonds. So just like when we talked about water and hydrogen bonds, these long chains, these of polypeptides, also have the ability to fold or to twirl and to bond with other ones in that long chain and form one of two types of secondary structures. One is the alpha helix. Helix is like the spiral staircase we were kind of talking about with DNA. Or it could be a beta pleated sheet, which is like the pleats of a skirt. So you get hydrogen bonding, but it looks a little bit different. So let's take a look at them. So up here you can see secondary protein structure. The beta pleated sheet is like this, where you get hydrogen bonding that occurs and you get kind of a pleating effect on it. And then the alpha helix, when it occurs, when you get hydrogen bonding here, instead of being this type of a pleat, it looks like a spiral. And within one protein, one very long set of amino acids, you can get both of these. So parts of it will form these pleated sheets, the beta pleated sheet, and some will form an alpha helix. So we're going to see more of that. Now, what is actually forming the hydrogen bonds is between some of the carbonyl groups. Remember what carbonyl groups are? We just looked at that with the carbohydrates. And some of the amino groups that are in close enough proximity that they start to form these hydrogen bonds. The third level, tertiary, so tertiary means third, so we have primary, the string of amino acids joined by peptide bonds. Secondary, we have either the beta pleated sheets or the alpha helices formed by hydrogen bonds. And now we have the tertiary. Now tertiary is the next level. So those first two happened first, and now this one would happen. And what this does is it really changes the shape of this protein, this polypeptide. And what it is, it's due to the interactions between various R groups that are on each of these amino acids. So we're going to look at R groups again, but the R groups now are going to interact. Some of them are going to be attracted to each other, some are going to be repelled from one another, some are going to form bridges like I was showing you earlier between sulfur, that's called a disulfide bridge. And the ultimate result is that you get this kind of more of a crazy looking shape here where you have certain amino acids. So this red is all representing a long peptide chain that has done some of it's a primary structure, and there's been already some secondary pleating or helices here, but now we have more. Now we have some of the amino acids on this side are going to interact maybe with an amino acid on the other side. Right here you can see there's an ionic bond that formed here. Where here you have something between these two, which are going to create something that's hydrophobic. So notice that this joining, this interaction between amino acids on this chain are causing these particular hydrophobic areas to go inward to try to get away from the watery environment. And then you have some others. Here's the disulfide bridge again. And here is another hydrogen bond that's forming here. So all of these things can happen in the tertiary structure. So it's the third level of protein forming these particular shapes. Then some go on to form larger structures which have quaternary structures. And with that, it means that we're going to get several of these things here can interact with each other, and we can get something that looks kind of more sort of globular. So you're going to see what that'll look like. It'll have something called subunits, where you might have, in this case, here, let's say that this tertiary structure that you're seeing right here, then is going to be represented right here. So look at the left here, where it's kind of lighter. And then there was another one that was had gone through primary, secondary, and tertiary that was sort of a darker looking one, and now here it is. And these two together are forming something that's called the quaternary structure. The last thing to mention on this, so ideally everything works perfectly. We get the right amino acids in the sequence, we get the right secondary structure, we get the tertiary structure, and then in some cases we'll get the quaternary structure. And then voila, what we have is a protein. And that protein is going to be able to do lots of different things. But there are things that can interfere with what a protein can do, and that's where we get into things that can do what's called denature a protein. So when a protein is in an environment that can cause its shape because some of those interactions between amino acids get changed in some way, they get broken, then, or perhaps just temporarily broken, and that's because of the conditions that are around it, we call that to be denatured. Things that can denature proteins are things like the change in the acid or base levels, the pH, or maybe changes in temperature in the environment. So these things can change the shape of the protein, and that can alter something with its structure. So if it's temporary and things can go back, then that protein is just temporarily unable to function. It can be changed back. But if it's a serious denaturing, where it actually breaks down the primary structure of the protein, then it can't go back to the way it should be, and that is being irreversibly denatured. So an example here is this fried egg. The egg white is protein. When you put it on in a skillet and it's hot, it's going to cause those long bonds of the primary structure to break down and kind of unravel. And so the egg turns from that clear, almost transparent look from the proteins that are in that shape to something like this, which is opaque, and it can't return to its normal state. So this egg, the egg white, is what we're talking about, has been denatured. It's been irreversibly denatured. It can't go back. And that's the end.